Hello everyone, I'm Dr. Sumedha-Ojha from Ashoka Institute of Technology and Management, Varanasi. Today I'll present a topic on genetic engineering, which is cloning and expression vectors. So, first of all, we'll talk about cloning vectors. Cloning vectors are those DNA molecules which are used to carry foreign genetic material into a host cell where it can be replicated. Basically, the cloning vectors are used to increase the copy number of the um, our genetic material that is of our interest. Okay, so there are several types of cloning vectors like plasmid vectors, bacteriophages, artificial chromosomes, etc. The characteristics of cloning vectors are they should have an origin of replication. And origin of replication is the point where the replication will start. It must have selectable marker genes. The selectable marker genes like antibiotic resistance genes, ampicillin resistance, kanamycin resistance, tetracycline resistance, etc. They must have multiple cloning sites. The multiple cloning sites have the recognition sites for multiple restriction enzymes. They must be small in size so that they can be easy to manipulate. They must have high copy numbers. High copy numbers ensure high yields of cloned DNA. They should have reporter genes. They are used to visually confirm the insertion of foreign DNA. They must have stability in the host to minimize recombination events or loss of the vector during cell division. The examples of cloning vectors include plasmids. The plasmids are circular DNA molecules commonly used in bacterial transformation. Bacteriophages. Bacteriophages are based on viruses that infect bacteria. Plasmids. They are hybrid vectors that combine features of plasmids and bacteriophages. Artificial chromosomes. They have bacterial artificial chromosomes, yeast artificial chromosomes, etc. Expression vectors are specialized DNA molecules designed to ensure the expression of a specific gene or DNA sequence of interest in a host chromosome. Here, these are used for expression of a gene. That means the gene has been transcribed and translated to protein. Okay. The characteristics of expression vectors include promoter sequence. They should have a promoter sequence for helping in transcription of the inserted gene. They must have transcription termination to terminate the transcription. They must have ribosome binding sites, which ensures efficient translation. They must have selectable marker genes. These selectable marker genes facilitate the identification of cells that have successfully taken up the vector. They should have multiple cloning sites for easy insertion of gene of interest. They should have protein tags for purification. This is optional, and they should have host specific elements like um, so that these host specific elements are very useful for expression in a particular host. And then they should have inducible or the repressible expression. These can be controlled. Replication of origin. This ensures the autonomous replication of the vector. They should be stable and should have an optimal copy number for expression. These are some of the examples of expression vectors. Um, in prokaryotic system PET series, these use T7 promoter for high-level protein expression in E. coli. Eukaryotic systems PCMV vector, these contain cytomegalovirus promoter for high expression in mammalian cells. Viral vectors, these are used in mammalian systems for high efficiency gene delivery and expression. I hope this video was informative for all of you. Thank you so much.